edit distance between the two DNA sequence or any two strings can be defined as a minimum number of operations that can be used to convert one DNA string into the other, where the edit operations are insertion, deletion, and substitutions. And we can convert the one DNA into the sequence of the other DNA. Or in case of strings, we can convert the one string into the sequence of other string. So in this case, we will use the edit distance technique and we will convert the sequence of TGCA-TAT and we will transform or convert it into this sequence ATCCGAT. And in order to convert this sequence into this sequence, we will use 5 editing operations. So first we will write the sequence of the DNA, that is this sequence, and first step will be we will delete the last T. So the last T is this one, so we will delete the last T and we will come up with the TGCATA. So in the next edit distance or editing operation, we will delete the last A. So this is the last A. We will delete this nucleotide and we will come up with the TGCAT. Then in the next step, we will add adenine, means A, at front. Front means the start of the sequence. So we have added A in the start of the sequence and then it will become ATGCAT. So in the fourth step, we will substitute C for G. So where there is a G, we will substitute it for C at position number 3. So the position number 3 is 1, 2 and 3. So this is 3 position and there is a G here. So we will substitute it with C. So we will add C here. So in the last step, we will insert G before the last A. So we know that the last A is this one. So what we will do, we will insert G before here and we have inserted G here. So if we see it very closely, we will see that we have come up with the same sequence as this is ATCCGAT, and we have again reached up ATCCGAT. So we have started from here and we end up here and we transform this sequence into this sequence, that is this one. So by using many editing processes, we have come up with this, and this is called the edit distance. So we have seen in the previous that we use the 5 steps to transform this sequence. This is the same sequence and we transform into this sequence, and we have used 5 steps. So in the previous slide, the edit distance between this and this was 5, but still we can use a better editing operations and we can transform this sequence into this by using only 4 editing operations. So again we can see here, this is the starting sequence that we have to transform into this sequence. The first step will be, we will insert A at front. So this is the front position, we have added A. Now it will become ATGCATAT. The next step is to delete T at position number 6. So position number 6 is 1, here 1, 2, 3, 4, 5, 6. So we have to delete T from this position. And if we delete this one, the sequence will become ATGCA, and T was here, it was deleted and become AT. So the third step will be the substitution of G for A at position number 5. So position number 5 is 1, 2, 3, 4, 5. So at 5 there is a nucleotide and we will replace it with G. So if we replace it with G, we will come out with this sequence AT, GC, GAT. And the last step is substitution of C for G at position number 3. And position number 3 is 1, 2, 3. 3 G and we will replace with C. So again we can see we have come up with the sequence ATC CGAT, that is this one sequence. So in this we have used only 4 steps to convert this sequence into this sequence. So we can say the added distance between TGCATAT and ATCCGAT is 4. So the less the added distance, better is the solution of the problem.